Welcome back to Studio Students. In this video, we'll talk about intestinal trematode infections. What do you mean by trematodes? Trematodes are nothing but unsegmented leaf-like flat forms, as you can see in this image. They are commonly known as flukes. Now that we know what trematodes are, let's classify them according to the organs they infect. Trematodes can be classified into intestinal flukes, blood flukes, hepatic flukes, and lung flukes. The important examples are Fasciolopsis buski for intestinal flukes. For blood flukes, we have Schistosoma. For hepatic flukes, we have Fasciula hepatica. For lung flukes, we have Paragonimus westermani. Most adult trematodes are flattened and leaf-like in appearance, as you can see in this picture. However, schistosomes are an important exception because they are elongated and cylindrical rather than leaf-shaped. Let's look at another important characteristics used in diagnosis, that eggs. The eggs of most trematodes are rubiculoted, meaning they possess a lid-like structure through which the larva emerges. Schistosome eggs are again an exception. They lack an operculum and instead possess a characteristic spine. To understand their life cycles, we first need to understand their larval stages. The first larva stage is Miracidium, which is a free-swimming larva, as you can see in this picture. The second stage happens in snails, which is known as Sporocysts. It has a sac-like body containing embryos. The third stage is Redia. In here, pharynx develops and the developing Cercaria develops. The Redia then develops into Cercaria, a motile larva with a tail. The Cercaria eventually transforms into Metacercaria, an encysted larva stage. Important point in here is that Schistosoma have only three larval stages, which include Miracidium, Sporocyst, and Cercaria. In most trematode infections, humans serve as the definitive host. A freshwater snail acts as the first intermediate host. For second intermediate host, we have either aquatic plant or crab. The infective stage for humans develops in this second intermediate host. For most nematodes, the infective stage is Metacercaria, except in Schistosoma. Humans acquire infection by consuming contaminated aquatic plants, fish, or crabs containing Metacercaria. After entering the body, the larva migrates to their preferred sites. The larvae migrate to the body and settle in the specific habitats. Some species inhabit the intestine, others the lungs, and some reach the liver. After maturation, adult worms begin producing eggs. The eggs produced by adult worms leave the body through feces or sputum, depending on the site of infection. Once these eggs reach water, the next stage of the cycle begins. In fresh water, the eggs hatch and release Miracidium. This free-swimming larva actively seek out and penetrate freshwater snails. Inside the snail, further development occurs. Within the snail, Miracidium develops into Sporocyst, then into Redia, and finally into Cercaria. The Cercaria are then released from the snail. The Cercaria escapes the snail and then infects aquatic plants or crabs. Then it develops into Metacercaria larva, which is infectious to man. Then man eats the crab and the loop goes on. Now let's talk about the life cycle of cystosomes. They require only freshwater snail as its intermediate host. Unlike other trematodes, the infective stage is not Metacercaria, but Cercaria itself. This difference is extremely important for examinations. Let's see how humans become infected. Humans acquire Schistosomiasis when Cercaria directly penetrates the skin during contact with contaminated fresh water. No ingestion is required. After skin penetration, the parasite begins its migration through the body. Following skin penetration, the larva enters blood vessels and travels through the circulation. Their first major destination is the lungs. From there, they continue their journey towards the liver. After reaching the lungs, the larvae enters into systemic circulation and subsequently portal circulation. They eventually reach the liver sinusoids, where maturation into adult forms occurs. The adult forms then migrate to the final venous habitats. Adult forms settle in specific venous plexuses depending on the species. It can be either in intestine or kidney and urinary bladder. Fertilized females lay eggs in this venous plexus, then it gets passed in feces. They mature in water and hatch as Miracidium larvae. The Miracidium develops inside the snail. It then develops into Sporocyst, then into Cercaria larva. It then leaves the snail and enters the fresh water again, which is infective to man, and the cycle continues again. The two important species of Schistosoma are Schistosoma mansoni and Schistosoma japonicum. Schistosoma mansoni is common in Africa, whereas japonicum is common in China. Let's begin with Schistosoma mansoni. The earliest clinical symptoms of the infection is cervical dermatitis. Skin penetration by Cercaria causes an itchy maculopapular rash at the site of entry. This represents the body's immediate response to invasion. Within 4 to 8 weeks, some patients develop acute schistosomiasis, also known as Katayama Fever. This occurs due to immune complex depositions and resembles serum sickness. The symptoms are systemic and reflect an exaggerated immune response. Chronic schistosomiasis results from eggs becoming trapped in tissues. These eggs induce granuloma formation and fibrosis. Complications include portal hypertension, pulmonary hypertension, neurocystosomiasis, and nephrosclerosis. Diagnosis is based on identifying characteristic X. The diagnosis of Schistosoma mansoni is commonly made through stool microscopy. The characteristic egg possesses a prominent lateral spine, as you can see in this picture. The egg cell also demonstrates acid-fast properties with modified satin staining. Additional tests can help detect active infection. Antigen detection such as ELISA can identify circulating cathodic antigens and circulating anodic antigens in serum and urine. The treatment of choice is Praziquantel, and ozamniquin is an alternative in certain cases. Let's now discuss Schistosoma japonicum. Schistosoma japonicum is more severe and has higher egg production. It is associated with colorectal and liver carcinoma. The eggs are smaller, rounder, and have rudimentary lateral spine, as you can see in this picture. The treatment is same as before. Fasciolopsis is caused by Fasciolopsis buski, which is the largest intestinal fluke infecting humans. Humans and pigs are definitive hosts, and snails and aquatic plants are intermediate hosts. The infective stage is Metacercaria. Clinical features include intestinal trauma, diarrhea, malabsorption, and yellow-green stools. The egg is large and operculated. The treatment of choice is praziquantel, and the alternative drug is niclosamide. And with that, we come to an end. Thank you. I hope you understood this video. If you like this video, please consider subscribing to my channel.